All right, so we'll be moving on to the next speaker, which incidentally is me. My name is Varun Dwaraka, and I serve as the Director of Research and Principal Investigator at True Diagnostic. I'll be very honest. This talk that I'm about to give, I've never given it. In fact, some of the results I've actually gotten about last weekend. And so it's fresh off the press, and I would love to get any feedback as well in terms of the results. But for us at True Diagnostic, we started off as a biological age testing company where we're leveraging epigenetics as a way to look at human health. But incidentally, through this endeavor, we've also been able to generate specific predictors on, for example, methylation risk scores of different diseases, but then also observing ways that we can maybe look at companion diagnostics. And so that'll be kind of my talk today. Is first of all going through and evaluating some of these epigenetic clocks that we develop and how they are used to predict drug response in HIV patients, specifically looking at a case study with GLP-1 agonists. And so with this said, this is the disclaimer, the usual disclaimer that we put out here. For disclosures, I do represent a corporate entity called True Diagnostic. I am the Director of Research, formerly of Bioinformatics. I also serve as faculty at the Geneva College of Longevity Sciences, Director of Epigenomics at the American Board of Precision Medicine, and have a few advisory roles at different supplement companies. But more importantly, what I'm trying to convey today are these particular learning objectives. There's going to be 4 parts to this talk. The first one is just a 101 of GLP-1s and semaglutides. Now, I imagine that if you're here, you're probably very well aware of these, so it's more of me trying to explain to myself what these things are, mainly because my background is in epigenetics and not really in semaglutide. So I'll kind of go through some of those mechanisms of action. Part 2 is going to go into the gerotherapeutics of this all. There's been some recent evidence to potentially indicate that GLP-1 receptor agonists can actually be used potentially for a longevity drug. Part 3 will go through that randomized control trial, and then part 4 is where are we headed. This is where I'm going to go through some of the very, very recent research on how we can leverage epigenetics as a way of figuring out if you are actually going to respond to a GLP-1 predictor or not. And it's a very similar type of concept that can be used in statins as well. So number 1, understanding GLP-1s and specifically semaglutide. So these GLP-1 agonists are essentially incretin hormones that are secreted from the L cells of the distal small intestine. One of the things that I found fascinating is that it's looking at this particular, looking at the particular schematic right here, it's not just expressed in the small intestine, but it has GLP-1 receptors are actually all over the human physiology of these different areas. So, for example, pancreatic B cells are these receptors are expressed there in the hypothalamus, gut, heart, kidney, and vasculature. And so the major actions of this is as glucose-dependent insulin secretion increases, the glucagon decreases as well. And ultimately in this interesting dance, if you will, it slows gastric emptying and acts centrally to reduce appetite and increase satiety for individuals who are on these agonists. And so what's interesting is that even though this was originally used for weight loss, really, there's been a lot of other effects that are being, especially for cardiovascular and renal effects, specifically with blood pressure reduction. I'm highlighting a few of these manuscripts that I think were very good reviews on actually looking through what these GLP-1 agonists are doing. So I think these slides are available, so I really urge you to look through some of those reviews. And currently, we have various different receptor agonists that are currently FDA approved, all the way from liraglutide to some of those semaglutides that are very popular nowadays, even in popular science, specifically Ozempic, Rybelsus, and Wegovy. There's multiple indications. So, for example, liraglutide, which was developed early on in 2010, has been used for obesity and overweight. However, there's multiple indications of semaglutide specifically where it's been used for type 2 diabetes and also for obesity as well, and some recent evidence to suggest with cardiovascular disease management. And the way the semaglutide specifically works is as a GLP-1 analog, the albumin binding potential for semaglutide allows for having a half-life of about 1 week, which is why you have at least a once weekly injection, especially if you're using Wegovy or Ozempic. This is actually from an expert review of clinical pharmacology, a fantastic review. I urge you to look at that. And it's been used in type 2 diabetes, maintenance, obesity, overweight with comorbidities. The SELECT trial, which was published, I think, in New England Journal of Medicine, showed that the usage of semaglutide has beneficial modulation of cardiovascular risk disease and also with liver disease as well. Specifically, the STEP trials, if you will, have been conducted. So STEP 1, 6, 3, 4, 5, 2, and 8 have already shown weight loss and metabolic effects, so the efficacy of semaglutide. The reason why I really like this image is that it actually shows the percent reduction in change in body weight compared to a placebo. So these were all placebo-based trials as well. And one thing to also mention is that the red bars are going to be semaglutide, the blues are the placebos, and then also liraglutide was included in step 8 as well. This suggests that semaglutide is actually a very efficient GLP-1 agonist, and one of the reasons why we've been focusing heavily, not only at our company, but just generally in the space, why semaglutide has been a part of many of these trials. And so what are the trials showing? I'm pretty much just highlighting some of the high-level findings, if you will. I imagine that people here are very well aware of the potentials. But especially in the step 1, what they're showing is at least a mean loss of about 14.9% reduction in overall weight. And then across that, we also see high rates of reversion from prediabetes to normal glycemia as well. And so there's been some improvements, essentially, on just looking at diabetic changes, especially with type 2 diabetes, and then also cardiometabolic risk factors being reduced as well. This particular trial right here was shown in Chin et al. 2023 for diabetes, obesity, and metabolism management. And so this paper actually goes through and looks at all of the recent results, if you will, from the STEP trials, if you will. There's also been improvements, especially with cardiovascular and kidney health as well. The SELECT trial is one of the premier trials that actually showed this, where in people with overweight, obesity, and established cardiovascular disease, but without diabetes, semaglutide actually reversed major adverse cardiovascular events by 20% relative to placebo. Ultimately, is showing that these semaglutides can be used in this conjunction as well. This is actually from the New England Journal of Medicine and Linkoff et al. 2023. Actually, it was a fantastic read. I know I'm a little bit of a nerd that way, but I urge you to really look at this particular trial as well. And then last but not least, we're also seeing them being used in specific liver diseases as well. So with a lot of these non-alcohol-based fatty liver diseases, we're seeing improvements, especially with resolution in these phase 2 trials. I'm not going to go too much into it, but the one that I really want to specify here is the SLIM liver trial, which was published in the annual Internal Medicine journal. The reason why I'm highlighting that trial is because we actually leverage some of that data in order to be able to develop and look at these epigenetic markers and how, specifically with epigenetic clock and these biological age clocks, how we're seeing reductions, especially with those individuals that are on semaglutide as well. So now, with all of that in kind of the back end, just as an introduction, part 2, I really want to make the case for why GLP-1 receptor agonist drugs, there's been a lot of talk about these as gerotherapeutics. And so to do that, let's define what a gerotherapeutic could be. The definition that came about very recently was an intervention that slows or modifies the rate of biological aging and thus delays multimorbidity. Some of the evidence to suggest that maybe these GLP-1 RAs can actually work on that is basically based on those trials that I alluded to. what we're seeing is that there's improvement in multiple risk factors, including obesity, glucose, blood pressure, inflammation, and reduce those cardiovascular kidney events. There was a recent editorial from Nature Biotechnology which actually went through the recent ARDD event, where that conference is all about anti-aging and just aging therapeutics and the biotechnology that goes around with it. The two keynote presenters were actually from Novo Nordisk and Eli Lilly. And one of the things that came from it is suggesting that these GLP-1s, especially during that talk, the GLP-1s function not just as weight loss drugs, but as a system-level perturbation to the aging network itself. So by modulating a lot of these age-related diseases, there's been a school of thought that suggests that you could maybe potentially start using these GLP-1s in the longevity space. Now, that's a very inflammatory statement, and I recognize that I might have lost a lot of street cred by saying that, but this is where we're starting to see that even with some of the trials and some of the evidence I'll probably show, that there might be a case to be made for it. Obviously, we'll have to do a lot more research. Now, when it comes to these biological age measures, so I've been talking about epigenetic clocks. I will define what those are momentarily. But one of the things to think about is, why would we even expect this in the first place? What is it about GLP-1s that we're thinking about that would even indicate that there might be improvements? Well, if you think about the research, especially in the aging space, a lot of it actually comes from caloric restriction. Especially with a lot of the interest, there's no work using validated aging biomarkers, such as epigenetic clocks or proteomic clocks, to examine these GLP-1 effects. And so, but there's been research actually on caloric restriction trials that show that many of these epigenetic predictors or these biological aging measures do decrease. Two that I actually highlighted here, the left one by Waziri et al. 2023 is actually one of my favorite studies to look at. It was in Nature Aging, where they used particular epigenetic clocks and showed that the blue line, especially right here, the blue line are individuals that have been consuming calories ad libitum, and those that are employing a 25% reduction of caloric intake. You could see the deviation between the overall biological age measure when you compare the two groups from essentially the control to the individuals that are employing a caloric restriction diet. We also see that on the right side, there's a paper that looked at telomere length, and that telomere length increases were observed as well in individuals that were actually undergoing a caloric restriction. So really suggesting that CR can modulate some of those classical aging biomarkers. And then this study actually, while I was preparing for this presentation, I ended up looking at this where it showed that it extends beyond caloric restrictions. Endurance and CR practitioners, they consistently show lower biological aging across many clinical and biological age measures, which include phenoage, DNA methylation, metabolomic, and microbiome-based bioage as well. So all of this is just to make the point that because caloric restriction slows down that whole biological aging concept, especially with these validated biomarkers, and GLP-1s induce similar kind of caloric restriction-based metabolic effects, maybe it's reasonable to hypothesize that GLP-1 therapy will also improve biological aging. And in fact, one of our collaborator, Michael Corley, recently put out that's a preprint, which is, I think, under review at NPJ Aging, on the slim liver trial. So took some of those samples from the slim liver trial and looked at individuals that were on GLP-1 versus those that are on placebo, and across multiple measures of biological age showed that there were significant decreases in the individuals that were on GLP-1 relative to the placebo arm as well. I will say this. a lot of the times these trials are being done on HIV-positive specific patients, mainly because they're using this as a way, GLP-1s are being used as a way to combat lipohypertrophy. And so as we go through, I will make that the limitation be that many of the data that we look at are on HIV-positive individuals, especially because you can't try getting a trial funded for healthy people on GLP-1s. It's very difficult, but it will be done soon as well. So in part 3 of my talk, apologies, one sec. So in part 3 of my talk, I'm actually going to go through and maybe explain a little bit about the relationship between epigenetic aging and semaglutide. I know I've been using these terms of these epigenetic clocks, and I'm not sure how many people are familiar with the space, but I want to be able to set the stage of what we're actually talking about here. So when I'm talking about epigenetic testing, I am not talking about genetic testing. This is not something similar to an MTHFR testing. We're not looking at genetic mutations in the in an individual. What we are looking at are the methylation marks that are on the DNA itself, specifically on the cytosine DNA methylation, cytosine base of the DNA. So the reason why this is important is because a school of thought is that these CPG sites or these cytosines, as they're methylated, can influence the gene expression patterns of a particular cell or individual, essentially. So the idea being is that if you don't have methylation marks on the CPG island or in a promoter region on those cytosines, technically the gene expression of that associated gene can increase, but then if it is methylated, it decreases. Now, that's the current paradigm. There's obviously, as anything in biology is there's also caveats to it, but that's the general paradigm of what we're doing. So when you do an epigenetic test, you are actually looking at the cytosine methylation marks and using that as a way to calculate what a biological age can be. Because you can calculate biological age using many different methods. This is one method to do so. And so what that ultimately comes back to is that these epigenetic clocks are essentially a measure in years where if you have a higher epigenetic age than your chronological age, that has actually shown associations to, for example, improvements in better health status. But individuals that have higher biological age may also show increases in cancer risk. For example, it can be due to stress as well. DNA methylation as a biomarker is very exposure-based, which is why this is actually a very good measurement of this whole biological age. And there's multiple being done here. So this is kind of my area of research. This is, if I had to have a zone of genius, this is kind of where I exist with majority of my research program. So with that said, though, when we recently worked with a group that had a RCT, randomized control trial, of a population of individuals, HIV-positive individuals, that had blood on a GLP-1 arm and a placebo arm. We wanted to just ask the basic question of, because of a lot of these caloric restriction based results that we're seeing with decreases in biological age, if an individual was on a GLP-1, would we see decreases in those biological age measures? You might be, and again, not to beat a dead horse, but why an HIV-positive group? Well, one of the reasons why is because this was the only set of samples that we were able to get access to. This was an RCT, and this is the gold standard of what we wanted to use in terms of being able to see the modulation of those epigenetic age. But if you also think about it, these people with HIV are a stress-tested model. We already know that individuals with HIV score higher in their biological age. And so if any kind of decreases that we see, we would hopefully see that with the GLP-1 arm and maybe not the placebo arm. And so it really becomes a good stress-tested model to ask, can a drug like semaglutide maybe even slow this acceleration? Because we already know that HIV will increase these biological ages. So with that said, this is a preprint. It's currently in review at Nature Communications. We just went through our first round. But in this 32-week randomized control trial, we had two different arms. One arm of 54 individuals were on semaglutide, this completely randomized and blinded. And then the other arm was placebos for 39 of those individuals. And they went through DNA methylation analysis. When they first went through this RCT, the primary endpoints were, the clinical endpoints were changes in body fat distribution, VAT scores, and metabolic parameters. So when we did this, these epigenetic markers weren't the primary outcome that we tested for. They were done ad hoc. So they were the secondary outcomes that we wanted to look at. And we looked at it based on the baseline and 32 weeks. So this was a 32-week trial with collections at 20 weeks. And we looked at multiple clock families. We developed a few of these epigenetic clocks, but the general space of epigenetics, there have been multiple of these clocks there. And why I'm mentioning that is because we had to use multiple of these epigenetic predictors just because each clock is tuned differently to another aspect of aging, and thus we wanted to see a comprehensive profile. And that's where it got really interesting. We didn't expect any of these changes because we just wanted to see what we would see from an exploratory analysis. But what we ended up seeing were, at least with 1, 2, 3, 4, 5, 6, 7 of these different predictors, we see significant reductions in the GLP-1 group of these individuals relative to the baseline. Relative to the placebo. On the right side, panels B, D, F, and H, you'll see is these are the relative changes of rates of those epigenetic clocks, the age rates. And so, my apologies. So this one right here is going to be the GLP-1 wing relative to the placebo. Hopefully that distinction is pretty clear just by looking at this analysis. So in 32 weeks, we're seeing about 2 to 5 year reduction in several of these validated mortality-based epigenetic clocks. Again, albeit this is an HIV-specific population, but with the placebo, we're hoping that we're going to be redoing this analysis, hopefully on some healthy individuals. We're hoping that this will translate there as well. So that's that interpretation. This study actually kind of went viral, even in its preprint state. And everyone and their mother was saying, "Oh, you should be on Ozempic. It decreases your biological age." And I had to take a self-care day because I was frustrated that that's not exactly what it's saying. It's saying that individuals on HIV, individuals that are HIV positive, show significant decreases, and we do need to do additional work to see if this is actually able to translate into a healthy population. But it is the first in-class evidence to suggest that semaglutide can modulate validated epigenetic clocks and typically affect some of these aging biomarkers. Now, the last part is going to be an interesting one, because my background is actually more in bioinformatics and machine learning and creating some of these predictors. And so the question now is, even from a corporate standpoint, what can we do with this information to help individuals with taking GLP-1s? And that's actually kind of some of the challenges, right? I've been talking to a lot of physicians at this conference, and a lot of it comes down to people don't know the dosing. They don't know if even GLP-1s are actually going to affect them. Is that even the right way to go? And so also the heterogeneity of the response as well. And this has actually kind of been solved by a lot of pharma companies. They have companion diagnostics. These are diagnostics that will help decide whether that particular drug that you're taking is right for you. And so that kind of concept is right patient, right GLP-1, right dose. And so this is interesting because actually it is a machine learning problem that you can actually solve by using some of the data, because from our original RCT results, what we were seeing is that individuals on the GLP-1 arm have an epigenetic signature that is different than the placebos. So can we leverage that information to actually create a predictor of whether an individual will respond well to GLP-1 or not? And so with that said, I kind of hunkered down in a basement for some time and tried to figure out how do we do this. And so a lot of this is actually based on the FDA cleared results that are already there. We already know that BMI, HbA1c, and HOMA-IR can be used, and essentially showing whether a person can be a responder or not. And so what I ended up doing was using some of these clinical outputs. I just pretty much asked, is this person a responder or not, based on the clinical data itself. So at baseline, 32 weeks, do we see this individual or these sets of individuals, are they responding well to this GLP-1, meaning are they decreasing their BMI by at least 5%? Are they showing improvements in their HSCRP, the inflammatory score of VAT, and then developing these responder statuses based off of that? Hopefully that makes sense. I'm happy to also take questions and maybe explain this a little bit more. But just having this responder and non-responder. Once you do that, all it is essentially a predictor. You can then use the epigenetics to predict the responder status using some of the baseline. So these scores on the right, the AUC scores, are essentially area under the curve. You can think of them as percentage of correct predictiveness. So meaning a AUC of 0.97 is that once we created the model and then put it back into a fresh new data set that it's never been trained, we were getting about 97% accuracy in correctly predicting if an individual was a responder or not. And this was also cross-validated, meaning it was run multiple times. So we were seeing anywhere from 88% to 99% accuracy in this. And I will say this is still in discovery phase. We're actually working with Eli Lilly on their tirzepatide program to actually validate some of this on tirzepatide as well. But this was really interesting because, again, we didn't have a huge data set. This was about maybe 200 people at most. With the baseline and post. And even with that small data set, we were clearly picking up an epigenetic signature that we're able to now leverage into predicting whether an individual would be responding to GLP-1 or not. The other thing, too, is it's not just whether a person will respond or not. We can also predict the percent change that we would expect based on their epigenetics, specifically with CAC scores, so coronary artery calcium scores. Those R-squared values are essentially what is the correlation of success in predicting, and we were getting 0.65 for some of these, which is a really good score for especially a low sample size here as well. So if you think about it in application, what we're trying to do is not only tell you whether an individual will respond or not, but we'll also predict how much of a response we would expect to see. And then in one of the other validations, again, this is where the RCT really came into play. Not only can we use this on the arm of individuals that are on GLP-1s, but we can also compare it onto the placebos. Because if, let's say, for example, you take the same predictor and put it onto a placebo, technically that AUC score, that chance of correctly predicting, should be 50-50. It should be just a random chance. And that's exactly what we're seeing, is that signature isn't something that's numerically derived. It's not just a byproduct of the numbers, if you will. It's actually a specific signature that is associated with GLP-1s and not with general metabolism changes. And so that's what is being shown on the left side is the AUC difference, the comparison between the GLP-1 prediction to just a random placebo group. And last but not least, this is more for my own edification. Because the RCT trial was showing that individuals on GLP-1 were significantly lower in their biological age, I wanted to just see, are these signatures actually being captured in the biological age clocks, or is this something completely different than these biological age measures? It is completely different. What you're seeing here is I actually created two versions of this predictor. One with just the epigenetic clocks and scores, and the other one is just using these CPG sites. And what you're hopefully you can appreciate is the level of difference in the AUC score between the clocks on the left and then just the general epigenetics on the right. So what this is suggesting is that, and it kind of sucks because some of the clocks that I helped develop seem like they're not actually capturing particularly the GLP-1s, but there's actually a much more of a deeper signal that we have to leverage. And so it's kind of it was an interesting argument, but it also goes to show that there's something a little bit deep-rooted in the metabolism that's being shown in the epigenetics specifically. So with that said, this is kind of the pathway where we're at. We're really on step one. We have a discovery. We identified that there's some type of a signature that is able to identify individuals that are GLP-1 responders or not. But how we have to move forward is going to step two, which we're currently doing, is seeing if this holds up in a healthy cohort of individuals. We've been working with individuals like Florence Comite and Larry Siegel with other individuals who are on or taking Ozempic, so healthy individuals that are taking Ozempic. We're working with Eli Lilly to see if a lot of these signatures also translate over to tirzepatide on their randomized control trials as well. And then we can then start, and if all of those things go according to plan, which they usually never do, but I'm staying hopeful, then we can start to actually develop these panels and then start to do prospective trials as well. But as of now, it's in its discovery stage. We have the availability of doing this, and we'll continue to work through. And I think this is where this epigenetic testing is going to move forward, is starting to make the case for whether individuals should be on a specific pharmacologic or not. I haven't even talked about our work with statins, because we've been able to do that as well on statin drugs as well, especially I think Pitavastatin specifically. I didn't have enough time to fit all that in here as well. So with that said, the summary and key takeaways. Part one, GLP-1. We went through just the background of all this. And then part two, hopefully there's some level of understanding that these GLP-1 RAs can potentially serve as gerotherapeutics. Obviously, we'll have to do a lot of randomized control trials and different trials just to see if that's even that claim can be actually backed up by science. And then hopefully in part three, our RCT analysis provides the first human trial evidence to show that semaglutide slows multiple epigenetic aging measures, especially in people with HIV, including mortality-linked clocks and pace of aging measures. And hopefully this future of this research is not only lucrative, but just exciting. How can we leverage epigenetics to be able to move this forward and to figure out individuals that are going to be positively impacted by a drug or not? And so with that said, I wanted to give a huge shout out to Michael J. Corley, who's my collaborator on this endeavor, and Ryan Smith, the founder of True Diagnostic, and actually one of my favorite people to work with on anything science and nerd-based. There was a lot of people at the booths as well. We actually have a booth here at the exhibit at 2024. I'll be there around 4:30 with the beer, so catch me then as well. I'd be happy to discuss more. But I think with that said, we're at time. I won't be taking questions just now. I would love to move on to the next speaker, but I'm happy to discuss anything and everything about epigenetics as well. So thank you so much for your attention.